so this is it. You're starting microbiology with basically me and Mr. Sakagnan on lectures and Madame Mouratou in the TDs. So how we organize things is kind of like this. So actually what I put in the slides on Madoc is from last year. This is the correct version. Okay, so we start off with me, and yeah. So I start off here. Next week we carry on. We have about 2.5 lectures on bacterial cell structure and function, and a little bit about taxonomy. That's about half a lecture. And then you start. We'll go on to look at bacterial trophic types, nutrition, energy metabolism, growth, and this kind of thing. And then I leave the place for Mr. Sakagnan, who will be teaching you about genetics, gene transfer, control of bacterial expression. And from week 43 onwards, this is just before the break, Madame Mouratou will start doing a kind of TD. In your timetable, all of these classes are noted as CTD, but in fact they're not really, okay? So we do the same thing as we do for the L2 microbiology. So me and Mr. Sakagnan give lectures, and there are TD that are supposed to illustrate what we've been talking about. So we pretty much do the same thing in English as we do for the other students in French. Okay, so after the break, Mr. Sakagnan finishes up his lectures almost. Then the last block of lectures is going to be with me, week 46 to 49, bacterial pathogens and viruses. We need to add one more TD session in week 49. So keep your eyes open on the timetable for that. And there will be a control, a test in week 50. Now, lab classes in the second half of the semester. Currently, one group is scheduled for Saturday mornings. Come here, please. You have to sit over there. So currently, one group is scheduled for Saturday morning, and I will try and change that before we actually have to do it, okay? So hopefully we can put that sometime during the week, from Monday to Friday, okay? So don't be worried too much about that. All right, and this part of the slide is just about what you can do or other courses you can study if you are interested in microbiology. So if you like this kind of subject, you can take the option virology, mycology, and parasitology in L3 next year. Afterwards, in Nantes, in the SPM master, there's one module that's microbiology. And in the M2 research, BBRT, there is parcours maladie infectieuse. So this is pretty much what we can offer you in microbiology in Nantes. Okay, another thing is, well, everything we're doing in this course is really quite standard, and it's well covered in microbiology textbooks. So, okay, come in, don't worry. Ooh, it might be difficult to find a chair. All right, let's have a look. Okay, they put us in a room with only 36 people capacity. So that's it. There are no more spaces, right? Did I miss something? Yeah. Okay, so let me introduce Charlotte Nancessy. She's come from Chester in the UK. She's on an Erasmus exchange. She's studying biology and French, right? So she's enrolled in the foreign languages faculty. but because she studies biology, she wants to follow a couple of classes in the science faculty. Okay, so that's why she's not on the list of the students, okay, in this group. So after, please make her feel at home and show her around the faculty or something. So I'm sorry about having no desk for you, but please take a seat. We'll try and follow this lecture. So that's why we have one extra person, okay? Right, so as I was saying, we're going to be covering pretty standard stuff during this course, and it will be in every microbiology textbook. So the ones that we have in the library, we have a few copies of Prescott, Harney, and Klein's Microbiology, which for me is basically the standard textbook for this course. And Perry's Microbiology also covers the same material. Okay, so if anything that's in the lectures you don't understand, you can go and check it out in these textbooks. Other things you might be interested in reading about is on some web resources. This one is a kind of free online microbiology textbook. This has got some teaching resources from the American Society for Microbiology. This one here. And this one is just more general interest. This is a kind of microbiology blog where people will write about the news in microbiology. So if you're interested, I recommend reading this one. Okay. So this is just kind of for reference. I don't really need to go through this slide right now, but this tells you what chapters to go to for the different bits of content that I'll be presenting. Okay, so if you need to look where to go, it's on this slide here. Basically, yeah, the other thing is here. The only time I'm really going to go beyond this basic textbook is some of the things in virology. So then you might need to go and look at a virology textbook for some things in the last lectures. Okay, that's about it for the introductory part. So now we're going to start working, really. Now, microbiology is what? It's, of course, the study of microscopic organisms. Anything you can't see with the naked eye falls into the domain of microbiology. So that means that microbiology is about bacteria, which fall into two classes, eubacteria and archaea. But it also includes microscopic eukaryotes, so fungi, yeasts, protists, and viruses. Now, most of this course, this is an introductory course, we're mostly going to be talking about eubacteria. Okay, you'll have one lecture from Mr. Sacagnon about the archaea, and you'll have a couple of lectures from me about viruses. But we're not going to talk about protists, yeast, fungi at all, okay? It's a little bit unfortunate, but I think we don't really have time to go into this subject. So most of what we're going to be doing is bacteriology, basically. Now, why are we so interested in bacteriology? Why is it worth spending time looking at these little guys? Well, one thing is that they're everywhere, okay? So once you start looking for bacteria in any kind of environment, you'll find some. On your skin, on the floor, out in the ground, in the dirt. That's perhaps not so surprising, but you can find bacteria anywhere. At the bottom of the ocean, living off molecular hydrogen. You can find bacteria kilometers underground in rocks. You can find bacteria living at 90 degrees in 1 molar sulfuric acid. They live in, they can live in a wide variety of places. So they're all around us. They're doing something in the natural environment. They're an important part of the living world. In fact, because they're everywhere, they're the biggest contributor to biomass on the whole planet. Now, most of the living world, in terms of mass, is microbes. Most of those are bacteria. So if you want to understand, life on this planet, you have to understand what bacteria are doing in those environments. Now, they can live everywhere because they can live on practically anything. They can generate metabolic energy from a huge variety of sources. Now, normally, in kind of most macrobiology, you think about animals, they're eating stuff, okay? We get our energy from eating organic carbon, or they're photosynthetic. And that's about it. But bacteria do more than that. They can get energy from a wide variety of chemical sources. And they can do that by mastering all sorts of clever biochemical tricks, transforming molecules from one kind to another and getting energy out of it. And that's helpful for us, because if we can understand how they can do that, then we can harness the kind of biochemistry that bacteria can perform and use it in industrial applications. Okay, at a simple level is something like, I don't know, producing cheese. That's an application in microbiology. But producing antibiotics, producing in biotechnology, producing recombinant proteins. We need to be able to understand bacterial growth and bacterial biochemistry to get them to work for us. That's kind of from an industrial point of view. From a basic science point of view, bacteria are the simplest living organisms. So if you're interested in the fundamental processes of life, DNA replication, transcription, biochemistry, this kind of thing, the easiest way to work on these processes is to look at bacteria, because they're the simplest model system. From a medical point of view, bacteria are responsible for a lot of disease. Learning how to control and treat bacterial infections implies that you have to understand how they work, how these bacteria grow. And a kind of other side of that coin is that they are not just pathogens with relation to human beings. Most bacteria that live in us or on us are not pathogenic at all, but they are commensal organisms or even in symbiosis with us. Bacteria in your gut are very important for normal gut function. Bacteria on your skin can also have an important function in regulating the immune response in your skin. Or through the skin. So more and more we begin to understand that, and if you remove these bacteria, then this has consequences for your physiology. That's from a very kind of human-centered view, but there are a lot of other very important elements of world ecosystems that are symbioses with bacteria. So nitrogen-fixing bacteria in plants, for example, have a very important role in plant biology. Okay, so bacteria are important at a lot of different levels. And an understanding of fundamental bacteriology will help a lot of different types of biologists understand how their systems are working. So that's why we're interested in bacteria. Now, what are the techniques? But, okay, before I get into really what are the structures of a bacterial cell and their different functions, most of that part of the lecture is going to be just me telling you it's like this and it works like that. Okay, but before we get onto that, I just want to introduce how we actually know all this stuff, all right? It's in a textbook, somebody wrote it down, but how do we know it's true? So what are the techniques you use for studying bacteria? Well, basically for bacterial structure, obviously looking at what kind of shape they are, okay? Microscopy, that's the most basic way of finding out something about bacteria. So you can sometimes see bacteria without specific stains with a phase contrast microscopy. Okay, so you can see basically the shape of the bacterial cell. Sometimes you can see the presence of inclusions in some types of bacteria. It gives you, so you get some kind of basic information. Now, depending on what microscope you have, this might not be very easy to see bacteria like this, and most of the time you'll be looking at bacteria after staining them. Now, stains can either be very simple, like crystal violet, or bacterial cells will be stained, or methylene blue. And once again, you don't get very much information. You see the size of the cell and its basic shape. More useful are differential stains, in which some types of bacteria will be stained and others won't. So the most common is gram staining, like here. So you see gram-positive bacilli here in purple, and gram-negatives, which are pink. This is telling you something about the nature of the cell wall, in fact. Another type of selective or differential stain is acid-fast staining. That's specific for mycobacteria. So the Mycobacterium tuberculosis. If you're coughing and spitting, you have night sweats, you're losing weight, you might get a test to see if you have mycobacteria in your lungs. And they'll use this test just to stain the bacteria specifically. Very specific for mycobacteria, gram-positives and gram-negatives will be washed, the stain will be washed out for them. So these pink things here, these are mycobacteria. So other specific stains can show up different kind of structures inside the bacterial cell, like endospores here. So what's red here are the spores, and the vegetative cells are blue here. So microscopy. And once you've got that, with gram staining, you can get some kind of basic ideas about bacterial structural types. So they're either round, coxy, or rod-shaped, bacilli, basically. So for coxy, everything in blue here, this is gram-positive, pink, gram-negative. So coxy, you can have monocoxy, diplocoxy, staphylocoxy, and streptocoxy. And for bacilli, you can have some kind of different shapes. Basilia kind of is a generic term for long rods, straight rods. Vibrio means anything that's curved. Okay. And coliform, it means it looks like Escherichia coli. It's a kind of short, ovoid kind of cell. Now, one thing that is a bit, or can be a bit confusing here, is that these terms, these are very general terms that describe the shape of the bacterial cell. But sometimes they are also used as part of the name of a bacterial species. Okay, so Streptococcus thermophilus, bacterial species. Staphylococcus aureus, bacterial species. Based on the name of, well, based on the morphology of the cells under the microscope. This one is confusing. When you say bacilli, that means rod-shaped bacteria. It could be gram-positive or gram-negative. When you say bacillus, that's part of a genus name, okay? Bacillus subtilis, Bacillus megaterium. That's always gram-positive, okay? Same thing with Vibrio is difficult because Vibrio could mean a comma-shaped bacterium, but it's also a genus name, Vibrio cholera, as a species of bacteria. So when it's written like this, not in italics, it's a generic term, just describes the shape of the cell. In italics, it's got something to do with the species name. Okay. One last thing to say about this is that the vast majority of gram-negatives are bacilli. Okay. Some gram-negative cocci do exist, often diplococcus with two cells that look like two coffee beans stuck together, and Neisseria, like Neisseria gonorrhea, Neisseria meningitidis. That's the most common gram-negative cocci. Oh yeah, last thing I was going to forget. You can't classify all bacteria as gram-positive or negatives. Okay, exceptions exist. Mycobacteria. So they're acid-fast. They're not gram-positive, not really gram-negative. Some bacteria don't have a cell wall at all. They also can't be classified. Okay, so that's light microscopy. What's the next step? The next step is using some kind of microscopy that has better resolution, electron microscopy. So you can use scanning electron microscopy. This is just going to give you an idea of the outside of the bacterial cell. Not very much more detail than light microscopy, to be honest. Or transmission electron microscopy. So depending on the technique, either you can take a section across a bacterial cell. For the first time, this is going to give you information of what is inside the bacteria. Okay, so you can see that, okay, we've got a kind of cell wall on the outside here, some kind of cytosol. This is the nucleoid, and then some inclusion bodies. You get an idea of what's inside the cell. With other techniques, you can kind of shade the surface and have an idea of a clearer picture or a more exact picture of what's on the surface of a cell. And here, for the first time, you can see that you don't actually have a smooth surface on most bacterial cells. That's what it looks like. You know, this looks pretty smooth, right? So it seems like there's nothing there, but it's just that you can't actually resolve the structures that are on the outside of the cell here. You need to have high resolution microscopy to see all these filamentous structures. So these are kind of what you would call pili, hair on the outside of the bacteria, and flagella. And you need electron microscopy really to see these things. That's one way to. So light and electron microscopy gives you some kind of idea about the structure of the bacterial cell. Another way to do things is a kind of biochemical method. You get a whole bunch of bacteria and smash them up, and then try and fractionate it into different things. So if you want to know what the cell wall is made of, you get a whole bunch of bacteria, smash them up, separate out the cell wall fraction, and then analyze it biochemically and see what's in there. So once you do that, you can find out that, for example, the outer leaflet of the outer membrane of gram-negative bacteria has got a lot of lipopolysaccharide, LPS. And this is its structure here. So you've got a lipid part and then a saccharide part up here. Another kind of approach to finding out what structures are in what part of the bacterial cell and what they do is to try and combine genetics and structural biology. So what I mean by that is, imagine you've got some kind of bacterium, grows very well on maltose or something like that, and you get some mutant, it doesn't grow on maltose anymore. So you can try and map the gene that is responsible, that is mutated, and that doesn't allow the bacterium to grow on maltose anymore, and find out what it does. So you can clone up this gene, try and express it, get the structure, find out where it sits in the bacterial cell, and you might find that it's a bacterial porin. This will sit in the outside of the bacterial outer membrane, and from the structure, and if you analyze the structure, you can see that you've got a trimer of this beta barrel thing here. So you've got to imagine the hydrophobic membrane is here, and these circles form pores where the maltose can get through. And so this combination of a kind of looking for mutants, mapping the gene, finding out what the gene does, and finding out the structure of the protein, this is going to give you information about what structures are performing what function in the bacterial cell. And once you put all this together, then you end up with models of quite complex structures in bacterial cells. So this is just an illustration. This is a current model of the basal assembly of a bacterial flagellum. A very, very complex nanomachine. This whole thing can spin round and round and round and round, like a rotor. Now, without going into details of what this does, the only thing I want to say here is that, you know, it's a computer graphic, but everything on here is to scale, and when you have each of these different colors, that means it's a different protein, and the lines between them means it's a different subunit. This is all exactly, it just really is like that. It's not just somebody just made it up and drew it out with his hand like that. It's really like this in real life. And the reason we know that is because people have been working on this problem for years, maybe decades. It's an accumulation of information that allows us to build this kind of model and understand, well, how bacteria do different things. Okay, so everything that I'm going to present to you now is, what we is a result of all these, decades of research. Okay, let's have a little break and see who's been listening. Okay, so if you want to go on to respond.cc, I'll send you out this question and see. Yeah, so actually, I better just write the key here. Okay, so if you want to send me your answers, if you're not connected, just think about it. And this is the key. Maybe. Okay, I'm just going to carry on being far too hot. Oh, it's a good thing these answers are anonymous. Okay, so I've got a few answers. So let's see what the results are. Okay. Let's see what people said. Okay. Capsule, that's correct actually. I forgot to mention this, but actually you can see the capsule with a non-specific stain. On the methylene blue picture earlier on, you could just about see the capsule. So that's true. That's a kind of region outside outside the bacterial cell. It's visible with light microscopy. Okay, Pili, you're right. Invisible, too small. LPS, it's a molecule. You can't see that with a microscope. Endospores, yes, they can be seen. Endospore specific stain, differential stain for endospores. Anybody who said yes, you're right. Nucleus. Go back to the lycée. Okay, you fell for the trick question. Why am I? You fell for the trick question. We're talking about bacteria. Bacteria don't have a nucleus, do they? So this is the excuse for me to give you a reminder about the fundamental differences between prokaryotic and eukaryotic cells, right? So don't forget these, right? So basically, okay, in eukaryotes we have organelles, intracellular organelles that are membrane bound, Golgi apparatus, mitochondria, this kind of thing. In prokaryotes, generally none. No true membrane-bound organelles. There are inclusion bodies, and we'll talk about them a little bit later, but they're not really the same thing as a membrane-bound organelle. Cytoskeleton. Yeah, eukaryotic cells have got a cytoskeleton. It allows movement between these different compartments, and keeps the cell in a kind of flexible, manageable shape. Now, classically, people would say that bacteria don't have a cytoskeleton. But let's say within the last 15 or 20 years or so, it's been found that, in fact, bacteria do have a kind of very, very basic cytoskeleton with bacterial equivalents to tubulin and actin, which are these two things. We'll talk about them probably next week. Cell wall. Okay, all bacteria have a cell wall, with very, very few exceptions, made of peptidoglycan. Some eukaryotes do, but chemically it's very different. So peptidoglycan in the cell wall is typical of prokaryotes, and particularly eubacteria. Archaea don't really have this. External membrane, okay, all eukaryotic cells have just got a plasma membrane. Bacteria, 1 or 2. Gram-negatives have got an external membrane. So they have 2. Okay, external outer membrane. Pili, these hairs that you can see on the outside of the bacterial cell. Strictly bacterial. Yeast don't have them. We don't, amoeba don't have them. Animal cells don't have these things. Flagella. Okay, sometimes in bacteria, sometimes in eukaryotes, but again, not the same structure at all. These are not homologous structures. Capsule, not really in eukaryotes. Some bacteria have them. Spores, some bacteria, some eukaryotes. Again, not really homologous structures. Finally, big differences in the way the genetic material is organized between eukaryotes and prokaryotes. Okay, the biggest one is no nucleus in prokaryotes. Now, eukaryotes mostly diploid or go between haploid and diploid phases. Some haploid eukaryotes, I guess, exist. But prokaryotes are generally haploid. In fact, always haploid. Chromosomes, several of them, in general, linear in eukaryotes. Mostly in prokaryotes, one circular chromosome. So chromosomal segregation during cell division is a lot simpler in prokaryotes. You don't have mitosis and all this complicated stuff. Very simple. But apart from having one big chromosome, prokaryotes usually often have plasmids, small circles of DNA that can be present in several copies or several hundred copies inside the bacterial cell. And last thing is that DNA is associated with histones in eukaryotes, not in eubacteria at least. Now, these are generally true for all prokaryotes, but there are interesting exceptions. So many bacteria have 2 chromosomes, not just 1. Doesn't really get any more than 2. Okay, 2 chromosomes plus some plasmids, that's about it. And some archaebacteria, some archaea, have histones. Okay, the eubacteria, they have other proteins that are basic, that are used to condense DNA. Not just naked DNA floating around in the cytosol of a bacterial cell. There are proteins associated, but they're not really histone proteins. Anyway, archaea, some types of archaea have histones. Okay, so this is just a bit of a resume of all of that. This is what a bacterial cell kind of looks like. So going from the outside towards the inside, the outermost layer is going to be the capsule. Then you'll have, you have the pili or fimbriae on the outside, flagella floating around, then the capsule, then the cell wall, and then you get inside cytosol, inclusion bodies, and the nucleoid. And what we're going to do is look at these structures. The thing that you need to understand really is what their functions are, what is their molecular structure, and have some idea about their biosynthesis. How do the bacteria make these structures? And we're going to start with the outside and go inside. Okay? So we're going to look at the cell wall first, then the capsule, and all these like fibers that can be on the outside of the bacterial cells, pili, fimbriae, and flagella. What are they? What are their different functions? And then we're going to look at the internal structures in a bacterial cell. Okay. Cell walls. What are their functions? Basically, you've got to have a rigid structure to the cell. Bacteria have a very, very rudimentary cytoskeleton. And the thing that really keeps them together is the cell wall that's on the outside. And this is going to protect especially against osmotic pressure. What will tend to happen is if bacteria are in an aquatic environment, there's a lot of water on the outside, it would tend to go into the cell. And if there's no cell wall, then these bacteria would just explode. They don't have like kidneys or stuff like that to regulate the amount of water in the bacterial cell. So you need something rigid on the outside of the cell just to maintain the structure. That's the function of the bacterial cell wall. And they're made of peptidoglycan, which we're going to see in a minute. Peptidoglycan is a polymer that is present only in bacteria, very, very specific to bacteria. That means that organisms that want to get rid of bacteria can produce things like lysozyme that we have, which can attack the bacterial cell wall, and other eukaryotes, like penicillium, can produce antibiotics that also attack the cell wall. And the other thing that's interesting for us is that among eubacteria, there are these two basic types of bacterial cells, gram-positives and gram-negatives, and they really differ in their cell wall structure. So before we go and look at that, the gram staining, how does that work? So when you do this in the labs, you'll carry out these different steps. So the first step is to use a non-specific stain, crystal violet. It's a basic dye. It's going to bind to everything that's acidic inside the bacterial cell. RNA, DNA, this kind of stuff. So it's all cells will be stained purple. Iodine is used as what is called a mordant. This term, I think, comes from the textiles industry. If you're dyeing cloth, you need a dye, and then a mordant will chemically fix the dye molecule to the cloth. So iodine here is going to fix the stain. Except when you wash the cells with alcohol, some of the cells will have the dye washed out, and other ones will retain the dye. And then the last step is counter staining with something that's a lighter color and will show up these cells that have had all the dye washed out from them. So fuchsin or fuchsin is the counter stain. Now the big difference here is that the gram-positive cells that retain the gram stain and are purple, they resist the alcohol washing step. Okay? And that's telling you that their cell walls are more resistant to solvents. Okay, one more thing to remember, don't forget, this is true about most eubacteria. There are some exceptions, mycobacteria acid-fast, they don't really work with gram staining. And there are some bacterial cells that are parasites on eukaryotes that don't have a cell wall at all. Okay, so the gram staining, this is telling us that gram-negative cell walls are more sensitive to solvents than gram-positive cell walls. Now, why is that? Well, if you're very, very lucky, you can perform some transmission electron microscopy and look at the structure of the bacterial cell walls. And if you look at something like this, is probably Staphylococcus. You can see it's part of a round cell. This is the cytosol. This is the plasma membrane, and then there's a big, thick layer of peptidoglycan. And that's it, okay? Plasma membrane, peroplasm here, and then peptidoglycan. Whereas in gram-negative cells, you have this kind of structure, cytosol, an internal membrane, a thin layer of peptidoglycan here, and then an outer membrane. So most of this cell wall structure is going to be lipid and not very much peptidoglycan. And that's why this kind of gram-negative cell wall is sensitive to the ethanol in gram staining. So the outer membrane can be washed out from the ethanol, and that's why all the dye can leak out. Whereas gram-positive cell wall, it's got a very thick layer of peptidoglycan. It's a little bit more resistant to solvents. Okay, peptidoglycan. What is it? This is the part of the cell wall that is rigid, gives a rigid structure to the cell. So what is it? Well, the clue's in the name, really, isn't it? Peptidoglycan. It's got a peptide part and a polysaccharide part. It's the glycan. So the polysaccharide part is a kind of repeating disaccharide of N-acetylglucosamine that is linked beta 1 to 4 with N-acetylmuramic acid. So this one is going to be called NAG, N-A-G, and this one is NAM, N-A-M, N-acetylmuramic acid. And this NAG-NAM beta 1 to 4 linkage is very specific in prokaryotic cells. So this is what's going to be destroyed by lysozyme. And breaking this link will break down the bacterial cell wall. Okay, so you can get an idea of, you've got these kind of, this repeating NAM-NAG, NAM-NAG, NAM-NAG polymer. This is going to give you lots of long strands of polysaccharide. Okay, these are sugars, basically. But that's not going to be very solid. It's going to be very rigid. It's just going to be like, you know, a load of hair or something like that. These are strands. These are fibers. In order for this to be, you know, solid in 2 or 3 dimensions, they need to be cross-linked. Okay, so these fibers are linked between them by the peptide part. So attached to each NAM, N-acetylmuramic acid, is a tetrapeptide. And the third amino acid of this tetrapeptide can be linked on to the last one of another one. So these are going to form cross-links between the polysaccharide chains of NAM-NAG. These are okay, the tetrapeptide and the interpeptide bridge. So these peptide bridges are essential for linking these strands together and giving rigidity to the structure. Okay. Now, tetrapeptide is very interesting. It goes like this. L-alanine, D-glutamine, diaminopimelic acid. Sometimes it can be lysine. And D-alanine. Now, there are two unusual things about these amino acids. Okay. What amino acids do you have in proteins? L or D? Think about it. Who says L? Hands up. Who says D? Who has forgotten all of their first year biochemistry? Everybody else in the whole class, yeah. In proteins, you only have L amino acids, right? The ribosome is only going to take L amino acids loaded onto a transfer RNA. But this thing has got some D amino acids in there. That's telling you straight away, this thing isn't produced on a ribosome, okay? Produced some other way. So you've got these unusual D amino acids. And this thing, diaminopimelic acid, what is that? It's not a regular, that's not a normal amino acid. So it's got a bit of an unusual structure. It's not the same as amino acids in proteins. Okay, so that's the structure. And I hope you can understand how it forms something rigid, okay? It's like Kevlar or something, bulletproof jacket for bacteria, right? It's quite tough. Now, how is it formed? And that's not an easy thing to do, right? Just imagine, okay? Ah. Now, these are the steps. Right, so I'm going to just give you the outline, then we can have a look at an animation that hopefully should illustrate this and make it clearer. Now, it starts off with making these subunits. The NAM-NAG disaccharide, together with a pentapeptide, that's synthesized in the cytosol of the bacterial cell, or rather on the internal face of the plasma membrane. Then, this whole kind of repeating unit, NAM-NAG pentapeptide, is flipped in the bacterial membrane and it goes out into the periplasm. So you make the repeating unit first inside the cytosol and you flip it onto the outer surface of the cell. And then you integrate it into the polymer. There's two steps. First, you've got to insert the NAM-NAG dimer into the polysaccharide part. So that's a transglycosidase that will be doing this. And then you've got to form the peptide bridges. That's a transpeptidase that does this. Now, before we look at the animation here, what you've got to think about is, why is this a big problem for the bacterial cell? Peptidoglycan, it's on the outside of the bacterium. It's a big, complicated polymer. When you synthesize a complex molecule, what have you got to put in order for it to work in biochemical terms or in thermodynamic terms in chemistry? What do you need? Got to put in energy, right? Think about like eating starch or something. We break it down, it gives you energy. To make it, you've got to put in energy. But this is on the outside of the cell. You don't have any ATP out here to give you metabolic energy. The ATP and all this kind of stuff is on the inside of the cell. So that's very tricky to synthesize these big structures on the outside of the cell. Right? So just bear that in mind when we look at this animation here. Okay, let's see. So this is one of the resources that is on the microbe library from the ASM. Now, just a bit of a reminder, this is what the cell wall looks like. You've got the plasma membrane here. The cell wall is on the outside, and it's peptidoglycan. Let's have a little bit of a zoom in here. So this is a gram-positive cell wall. No outer membrane, you've just got a lot of peptidoglycan here. So what's going to happen? Okay, it starts with UDP, N-acetylmuramic acid, and then the pentapeptide is synthesized on it. Then it's transferred onto this molecule here, which is bactoprenol. And then the bactoprenol, it goes too fast. He's going to do it again. Okay, so you start with UDP now, right? Pentapeptide is synthesized on it. Then you get the NAG, which is going to come on here. And then this bactoprenol, PP, NAMNAG, and the pentapeptide gets flipped over. And then the transglycosidases add this onto the NAMNAG structure here. And then the transpeptidases make the peptide bridges. So where does the energy come from? All right, you've got to, outside the cell, you've got to make two kinds of chemical bonds when you're synthesizing peptidoglycan. The glycosyl, the beta-1,4 linkage. Got to make that one. Okay, you made the dipeptide here, so you made one link that's in here. But this dimer has got to be added on here. So you've got to make one more link. This is a transglycosidation reaction though. You make one and you lose one. So it's even. Once you get outside the cell, you've stored up the energy. I wonder if I can go back to here. No, let's try and do this. Back to Pinot. Okay, this is the basic structure that's going to be flipped outside. So this is where you've got the energy to make the glycosidic linkage. This one here. So this gets broken down and it's transferred on to form the next glycosidic linkage. And the energy to make the peptide bond comes from here. What you synthesize in the cytosol is a pentapeptide. What you've got here is a tetrapeptide. So what you've done is you've cut off one of these amino acids. You use the energy from breaking this bond to form the new one, to make the tetrapeptide linkage. Okay? Transpeptidase does that. So that's why bacteria use this on the surface of it complicated system to form peptidoglycan, because it's really difficult to synthesize stuff on the outside of the cell. And that's why you form a pentapeptide first, and that's why you have this phospholinkage here, because it allows you to export the energy that you need to make the bonds outside the cell. Okay, so bacterial cell wall synthesis is the target of several different families of antibiotics. So I'm just going to go on to the next section here, which explains how they work. So bacitracin. This is an antibiotic. It's very toxic for your kidneys, so it can't be used as a systemic treatment, but you can get it in a kind of like cream or something for local infections sometimes. So bacitracin, what does it do, right? So here's your peptidoglycan synthesis. The NAMNAG gets attached to the bactoprenol diphosphate. The bactoprenol flips in the membrane. Okay, so this is a lipid transporter, but it prevents recirculation of the bactoprenol. So the cycle is disrupted. Okay, this can't be recycled and you can't export anymore. So it's going to inhibit cell wall synthesis. Okay, penicillins, cephalosporins, all of these beta-lactam family of antibiotics. They have a different mechanism of action. Okay, so it starts the same way. Okay, you get the bactoprenol diphosphate NAMNAG structure, flips in the membrane, and you need two activities, a transglycosidase and a transpeptidase. Okay, penicillins block the transpeptidase activity. So you can't form these crosslinks. Now, penicillins are bactericidal for bacteria that are growing. Okay. And that's because as a bacterial cell grows, it's got to destroy its own cell wall. Otherwise, you never have any free N-acetylmuramic acid to start putting in the new dimers. So as a bacterial cell grows, it's got to express what are called autolysins. They degrade the bacterial cell wall slightly, and then the transglycosidases and transpeptidases add new subunits, so the whole thing gets bigger. So if you block the transpeptidases, the addition of new subunits is inhibited, but the autolysins are still active. So little by little, they'll break down the bacterial cell wall and the cell will lyse. Okay? So there's two things about antibiotics. Sometimes they say they're bacteriostatic, they just stop cell growth, and bactericidal, they kill bacteria. Penicillins, cephalosporins are bactericidal, but only for actively growing cells. And the last one is vancomycin, okay, which has got a different mode of action. Once again, synthesis starts the same way. Bactoprenol, PP, NAM-NAG, transported over into the periplasm. This time, vancomycin is a large organic molecule. It's going to specifically recognize these two amino acids that are on the end of this pentapeptide. If you remember what they are D-alanine bound to D-alanine. So it's a very, very specific structure with two D-amino acids stuck together. You only find it in this pentapeptide. So that's why this antibiotic is specific. You know, we can take it, you can take it orally. It's not going to bind to any structures you have in your body. It will only bind to this, the final two amino acids in this pentapeptide. So it's going to bind on here and it will stop the transpeptidase from acting. Okay. Unfortunately, vancomycin is only active against gram-positive bacteria. For example, Staphylococcus. You can use it on Staphylococcus aureus infections. That's because gram-negative cells have got an outer membrane and vancomycin can't get across the outer membrane. It's too big. Whereas penicillins, cephalosporins, they're smaller molecules. They're active against gram-positives and gram-negatives. Just a question of the size of the molecule. Okay. Right. Oh yeah, I'm on the. That's why. I have to get back and take this out of here. Okay, so coming back to this. That's the structure of peptidoglycan, how it's synthesized. There are some differences in the structure between gram-positives and gram-negatives. So in gram-negatives, pretty much all gram-negatives, you have L-alanine D-glutamate, diaminopimelic acid, and D-alanine. So this DAP here, okay, the thing that you have to remember, it's got a free amine group here. And this amine group on the DAP can form a kind of peptide bond with the carboxylate group at the C-terminus of this D-alanine. That's the peptide crosslink here. That's what's formed by the transpeptidases in the cell membrane. And this is direct. Now in Staphylococcus aureus and some other gram-positives, you don't have a direct peptide link here. You have a kind of interbridge formed of a number of glycines. So what does that mean? Well, think about it, right? In gram-negatives, the peptidoglycan layer is very narrow. So all the strands you want to cross-link are very close to each other. However, in gram-positives, the peptidoglycan is much, much thicker. So if you've got this glycine bridge, that means that sometimes you can fix strands that are a bit further away from each other. And it helps this big, thick peptidoglycan layer to stay all together, okay, as one cohesive unit. Okay, the next part is the difference between gram-positive and gram-negative cell walls. Probably will finish up after that. Okay, gram-positives. Simple, right? They've got plasma membrane, a very thick peptidoglycan layer. The only other thing they have that's specific for gram-positives is teichoic acid and lipoteichoic acid. So these teichoic acids just means wall acids, right? They are polymers of, this is glycerol phosphate or ribitol phosphate. That's 3 carbon in glycerol, 5 carbon in ribitol. It will be the same kind of thing. So it's going to be a big linear polymer. And this R group will be glucose or an amino acid or another sugar. So it's a simple linear polymer with a lot of negative charge on it. So it's going to be covalently linked at one end to the peptidoglycan. And for lipoteichoic acid, the lipid part is going to be buried in the plasma membrane. And all along the length of the teichoic acid molecule, there are going to be electrostatic interactions between the negative charge here and positive charge on peptidoglycan. So this is going to help. What is the function of this? Is just trying to keep the different layers of the peptidoglycan all together. Okay, because otherwise, this is so thick that it's going to start like fracturing off. Okay, just think of it. You're building up layer on layer on layer on layer of this material, and you put some of this transversely to keep it all together. So gram-positives have that. Now, gram-negatives, more complicated. Okay, you've got plasma membrane, periplasm, peptidoglycan, and then an outer membrane. What's the thing to say about this first? So the outer membrane, okay, this is anchored to the peptidoglycan by something called Brown's lipoprotein. So that's the function of this one. It's in the outer membrane, and it's just there to make sure that they stay physically attached to each other. The peptidoglycan and the outer membrane stay physically attached through this lipoprotein. Now the other thing that gram-negatives, all gram-negatives must have that. All gram-negatives must also have porins. Because, I forgot to say this, okay, if you look back at gram-positives, peptidoglycan here, physically it's quite tough, but chemically it's porous, okay, so small molecules can diffuse through this peptidoglycan layer freely. Sugar, amino acids, antibiotics, they can all diffuse through here. All the stuff that's interesting for the bacteria can get here and then be imported into the cytosol. But in gram-negatives, the outer membrane is a barrier for diffusion. Okay, so if you have sugar coming in here, it's a hydrophilic molecule. It won't be able to diffuse across the lipid bilayer. So they all need porins, which will allow the diffusion of small hydrophilic molecules across the outer membrane, like sugar, amino acids, this kind of thing. They can get into the periplasm and then be imported into the cell. So all gram-negatives have to express porins in their outer membrane. The other thing that gram-negatives have on the outside, the outer leaflet, okay, the inner leaflet of the outer membrane, this is just ordinary phospholipid. The outer leaflet is lipopolysaccharide, LPS. Okay, what is LPS? Okay, lipo, that's easy. So it's got a lipid part, and the lipid part makes up the outer leaflet of the outer membrane. And it's composed of something called lipid A. That's the lipo part in lipopolysaccharide. The polysaccharide has got two different components of it. There's a core, which is highly conserved. You don't need to learn exactly what this is. You just have to remember that this core polysaccharide here is going to be the same between different gram-negative bacteria. It's conserved, the same in different species, or very few differences. Then attached to that, you've got a lot of repeated chains. So you'll have like 5 to 12 sugar molecules stuck together in a different way. And that 5 to 12 unit will be repeated several times, I don't know, dozens of times. So this part is going to be very, very long. And that's going to be very variable. Going to be different even between different strains of the same bacterial species. Yes? The O antigen repeat, that's going to be very long. Oh yeah, sorry. Don't. Okay, so this part is going to be very long. This is all conserved. This is very long and very variable between species. Okay, another word for lipopolysaccharide is endotoxin. And it's called endotoxin is because it can be recognized by parts of your immune system that are going to recognize this conserved part, in fact, and set up an acute phase reaction. So you have fever, diarrhea, septic shock. This is all because your body has recognized the presence of LPS, also known as endotoxin. That's something that's an effect on human beings, but that's not really the function of this stuff. Gram-negative bacteria don't have LPS just to kill you. They have it because it's useful for them. So it basically is going to protect the outer membrane from chemical attack. So for gut bacteria, they've got to survive being ingested and passing through your digestive tract. So bile salts normally would disrupt this membrane. They can disrupt fat globules, right? LPS will protect gram-negative bacteria from that. Also, it's going to prevent the diffusion of some antibiotics, okay, this LPS structure. However, it will help bacteria to protect themselves against from detergents. So it's chemical protection for the outer membrane. Last thing you need to know about this is that the O antigen, O is for O's, like sugars, because they are variable, they can be used for the identification of different bacterial strains, okay, below the species level, different substrains. Okay, now how is this synthesized? Actually, you don't need to know this for exams or tests because the other L2 students, they don't get this. But it's just for general interest, all right? Once again, LPS, it's a complex polymer on the outside of the bacterial cell. The problem for the bacterium is the same as for how to produce peptidoglycan. And in fact, there are some similarities. So the core oligosaccharide is synthesized on monophospholipid A in the cytosol. And then this whole thing is flipped over into the periplasm. Now the O antigen, the repeated O antigen subunits here, they're synthesized on, what could this be? Anybody have a guess what this is going to be? Similar problem to peptidoglycan? Similar problem, similar solution maybe? What is the NAM-NAG attached to for peptidoglycan synthesis that flips over? Bactoprenol. Okay, the bactoprenol. It's the same thing, okay? So this O antigen, the repeated subunit, is synthesized here, attached to bactoprenol, then it's flipped over. Just the same as the NAM-NAG subunits in peptidoglycan synthesis. And then once again, you've got the transglycosylation reaction here, which is going to add all these repeated subunits onto a growing polysaccharide chain. Then after a while, I don't exactly know how this is controlled. This polysaccharide chain here that's on a bactoprenol is going to be transferred onto the core subunit here, the core oligosaccharide. And then the whole thing, once it's synthesized in the periplasm, is going to be transported across the peptidoglycan and put into the outer surface, outer leaflet of the outer membrane. There's a whole multi-protein complex that grabs this and pushes it to the other side. Takes energy, but this energy is supplied on the internal face here. So it's in the cytosol where you have ATP. Okay, so that's how LPS synthesis works. Okay, let's have one question. I'm not going to send this off to MClickers because it might take a little bit of time. Just think about this. There's only one correct answer. We'll do it by a show of hands. And then we'll stop there. Okay, everybody thought about that enough? I'll take that for a yes. Okay, so who says answer 1? LPS protects gram-negative cells from lysozyme. Nobody. Who says B? Lysozyme attacks only the polyglypeptides that are present only in gram-negatives. Oh, that's wrong. Only in gram-positives. That's a mistake, right? This one is in Staphylococcus aureus. Only in gram-positives. Who says that's right? No. Who says this one? Lysozyme cannot cross the outer membrane. 1, 2, 3, 4, 5, 6, about 6, 7 people, 8 maybe. Who says this one? Lysozyme disrupts the teichoic acid. Okay, 7 people, you're right. Okay, it's too big. Lysozyme is an enzyme, it's a protein. No way is that protein going to diffuse across the membrane here. Can't get through the lipid. It's too big to go through the porins. Something that can come through here is going to be a few hundred Daltons in size, okay? Maltose, a disaccharide, couple of amino acids, something like that. Big proteins are not going to be able to get across the outer membrane. Okay, I think that's a pretty good time for us to stop, so we'll carry on next week on Monday. One more thing, because I still got 3 minutes that I kept on purpose. Right, what I like to do is once or twice a week, maybe after every lecture, is in the evenings have a kind of competition between you called Microbe of the Week. So what I do is tonight at 7 p.m. I'll send out an email to all of you with a question. All right, and you have to find out the answer and email me back what the answer to the question is, right? So what I give is like 3 points for the first correct answer, 1 point for every correct answer, and bonus points for people who write some interesting stuff. And don't just copy out what you find on Wikipedia. It has to be your own like composition. People who just copy and paste, that's very fast. So you can be first, but I don't count that, all right? So it's optional. It doesn't count for any marks. It's just for people who are keen, okay? But there are 2 good reasons for taking part in Microbe of the Week. First is there is a prize on the last session. Okay, so I keep score, and the person who's got the most points, they get a real prize. It's not money. It's not bacteria. It's something, okay? So there's a prize. Second point is, if you are interested in having an international mobility year next year, you have to ask me for a recommendation letter. And if I can write things like, this student is very keen, showed interest in the subject, it gives me something nice and positive to say about you, okay? So think about it that way. Okay, so 7 p.m. I'll send out the question. If you want to play, get ready and get sending back answers, okay? So see you Monday otherwise.